Today we are going to do structure and functions of hemoglobin, myoglobin, hemocyanins and hemorythrin. Normally मैं कुछ एक्सप्लेन नहीं करती हूँ, सिर्फ रीडिंग करती हूँ क्योंकि मुझे लगता है शायद मेरी जो राइटिंग है वो समझ नहीं आ रही है आप लोगों को। अगर राइटिंग समझ आ रही है तो आप लोगों को म्यूट करके सुन लेना आप लोग। चलो स्टार्ट करते हैं। हीमोग्लोबिन। The hemoglobin are globular proteins in high concentration in red blood cells that bind oxygen in the lungs and transport oxygen from the blood to the tissues and cells about the capillary beds of the vascular systems. On returning to the lungs from the capillary beds, hemoglobin acts to transport CO2 and proton. Structure. The structure of hemoglobin is classified under two headings, which is structure of heme, the prosthetic group, and structure of globin, which is known as the protein group. Heme, the prosthetic group. फोर पॉलीपेप्टाइड चेन्स होती हैं इन हेमोग्लोबिन एंड द सिंगल पॉलीपेप्टाइड चेन होती है इन द मायोग्लोबिन contain a prosthetic group heme. A prosthetic group is a non-polypeptide moiety that forms a part of a protein in its native functional state. A protein without its prosthetic group is designated as an apoprotein. The heme is a porphyrin molecule found in hemoglobin and myoglobin. The iron atom is the ferrous +2 charge oxidation state in functional hemoglobin and myoglobin. You have to make the structure of heme जो मैंने उसमें hemoglobin वाली वीडियो में करवा दिया है. The ferrous atom in the heme form 5 or 6 ligand depending on whether or not O2 is bound to the protein. Of the 5 or 6 bonds, 4 of these bonds are to the pyrrole nitrogens of the porphyrin. Since all the pyrrole rings of the porphyrin lie in a common plane, the 4 ligands from the porphyrin to the iron atom at its center have a tendency to lie in the plane of the porphyrin ring. The fifth and the sixth ligand bonds to the ferrous atom of the heme are directed along an axis perpendicular to the plane of the porphyrin ring. The fifth coordinate bond of the ferrous atom in each of the heme is to a nitrogen of a histidine imidazole. This histidine is designated as proximal histidine in the hemoglobin and myoglobin structures. Now what is globin? The globin, a protein part of a hemoglobin, is composed of 4 parallel layers of closely packed polypeptide chains which are called alpha, beta, gamma, delta and epsilon chains. The alpha chains have identical amino acid composition of 141 amino acids. The beta and gamma chains are made up of 146 amino acids and the total number of amino acids in globin is 574. C-terminal amino acid and N-terminal amino acid are here given in this structure. Hemoglobin molecule and its subunits contain mostly hydrophobic amino acids internally and hydrophilic amino acids on their surfaces. So they form heme pockets. Now myoglobin. Myoglobin of red muscle tissue stores oxygen under conditions of oxygen deprivation. This oxygen is released from use by muscle mitochondria for oxygen-dependent synthesis of ATP. Myoglobin, a single polypeptide chain of molecular weight 17,000 with the protein chain containing 153 amino acid residues folded about the single heme group. The surface is polar and the interior nonpolar residues both polar and nonpolar regions. X-ray Crystallographic analysis revealed myoglobin to be a compact, roughly and spherical molecule. Approx 75% of the residues are present in 8 right-handed alpha helicals starting at the N-terminals. These are termed helices A through H. In helical, interhelical regions are identified by the letters of the two helical regions they connect. When oxygen bonds to myoglobin, the bond between one oxygen atom and the Fe+2 forms perpendicular to the plane of the heme ring. The second oxygen atom bonds at angle of 121 degrees to the plane of the heme and oriented away from the distal histidine. Diagram shows preferred angles for bonding of oxygen of carbon monoxide to the iron atom of free heme solid line. Carbon monoxide bonds to isolated heme about 25,000 times more strongly than does oxygen. Functions of hemoglobin and myoglobin. Hemoglobins are structurally related proteins of vertebrated erythrocytes that perform two major biological functions, which are transport of O2 from the respiratory organ to peripheral tissues and transport of CO2 and protons from peripheral tissues to the respiratory organ. Myoglobin of red muscle tissue stores oxygen. Iron in hemoglobin and myoglobin. In hemoglobin and myoglobin, the Fe2 cation is coordinated to the four nitrogens of a protoporphyrin IX ligand. Without the molecule of oxygen, the Fe2 cations of deoxyhemoglobin and deoxymyoglobin are in a high spin state and the molecule are paramagnetic overall. Kinetics of hemoglobin and myoglobin oxygenation. Myoglobin stores oxygen in muscle tissue while hemoglobin transports oxygen from the lungs to the cells and then picks up CO2 and transport it back to the lungs for exhalation. The spatial properties of hemoglobin molecule that make it such an effective oxygen carrier are understood by comparing the O2 minus binding or O2 minus saturation curves of myoglobin and hemoglobin, which are the oxygen and which are oxygen binding curves of myoglobin and hemoglobin are given here in this graph. So here it is. Next, Hb plus 4O2, which is HbO2 whole 4. This shows the percentage of O2 binding sites of hemoglobin or myoglobin that are occupied by O2 molecule when solution of these proteins are in equilibrium with different partial pressure of oxygen. So thank you so much for watching hemoglobin and myoglobin. We will do hemerythrin next time. Okay, so thank you so much.